All right, everybody. This is Mr. Miller. So no guest speaker today. The cell cycle. Cell cycle is the life cycle of a cell. And this first video is going to sort of talk about it. It concentrates on one main part of that life cycle, because really this is kind of where all the action happens. This is the reproduction of the cell when it splits into two, because we have to get a copy of DNA into both cells. How does that happen? So that's a big part of it, but there's more to the cell's life cycle than that. So pay attention to the video.
Nearly every organism that is made up of many cells begins life as the single cell of a fertilized egg. That single cell divides over and over again until eventually an embryo is formed that is made up of trillions of cells of many different types. Over time, the embryo develops into a baby. And even at a very young age, some of the baby's cells begin to wear out. In fact, in a typical human being, every second of every day witnesses the death of about 50 million cells. Therefore, new cells must be constantly produced to replace old, dead, and damaged cells. For cells to reproduce themselves, whether in a developing embryo or in a fully grown adult organism, certain definite steps must be followed to assure that the new cells will contain exactly the same genetic material or genes that were originally present in the parent cell. And the essential process underlying the reproduction of cells is called mitosis. Mitosis is defined as the duplication and division of the nucleus of a cell and its chromosomes during cell reproduction. Scientists recognize four distinct stages of mitosis. First is prophase, second is metaphase, third is anaphase, and fourth is telophase. An average of about 6% of a cell's total lifespan is spent in these four stages of mitosis, while the other 94% of its life is spent in a stage that is not considered to be a part of mitosis, called interphase.
All right, a cell's life, the life of a cell, sounds like a book. The cell passes through different stages in their lives. These stages, just like we do, and we're actually probably going to compare a cell to a human life cycle. Just like humans, the cells pass through stages, and these stages are called the cell cycle. Begins when the cell first forms, just like us when we're born, but a little bit different is a cell splits into two, so it has two cells instead of just giving birth to a single cell. And it ends when the cell divides. So unlike our life cycle, the cell's life cycle ends when it reproduces, because it's splitting in half and becoming two cells, each sort of smaller than the original. Before division, though, it has to copy its DNA. All right, so in prokaryotic cells, we learned about prokaryotic and eukaryotic cells. Prokaryotic are bacteria. Eukaryotic cells are the cells in you and other complex organisms. Prokaryotic cells are very simple. There's no membrane covered organelles, so it's very easy for them to divide up the ribosomes into two different cells. And there's just a small circle of DNA. So it copies its DNA, it stays in a circle, and then it splits into two. So here's one copy of the DNA, another copy of the DNA. It divides in two. So you got one identical copy of the DNA here, one identical copy of the DNA here. And then it just splits using binary fission. And since it has a cell wall, it's going to have to build that cell wall and separate the two. It doesn't really look like it has a cell wall here, but they do. And then each cell has a full set of DNA, or a full brain. All right, eukaryotic cells, that's us. They're more complex. There's more DNA, and the DNA is in these long strands instead of a circle. The DNA is in chromosome pairs, and there's homologous chromosomes. We'll talk about that. We'll talk about what a chromosome is here in a second. Humans have 23 pairs, or 46 chromosomes altogether. We're going to talk more about this in the next unit, but the 23 pairs, we get one set or pair, one half of the pair from mom, one half of the pair from dad. So we actually have copies of the DNA, but not identical copies. And we'll talk about that in the next unit. All right, what do chromosomes look like? These are the identical copies. So this is one copy of the DNA, and this is the identical copy. And they connect together at what's called the centromere. This this holds them together so they can be separated. They're bundled up. This is when they're actually chromosomes. Otherwise, it's just loose in the chromatin inside the nucleus. But here they're actually chromosomes. So they're bundled up DNA. Identical copy, identical copy. They're attached here so they can be pulled apart. And we'll see how that happens in the steps of mitosis. Okay, the cell cycle. This is sort of an overview image of the cell cycle. You're going to be making a movie pretty soon of this. So you're going to start off with storyboards, which is sort of like a blueprint. You're going to want to consider what images you want to put in that blueprint that you want in your movie. An overview picture like this, there's other types where you could have like a cell, show the cell going through the different stages. Might not be a bad idea, but something like this where it shows the different phases so you can talk about an overview of the cell cycle. So you can see there's the replication stage, there's the growth and preparation. So replication and DNA replication DNA needs to be copied. Growth and preparation for mitosis. There's a big part here called interphase, and that's what you're going to be learning about next. Interphase is kind of the life phase where it's growing. The DNA copies itself at some point, but it's also doing whatever the cell does. If it's a muscle cell, it would be doing what a muscle cell does, contracting and doing what a muscle cell does. Then there's mitosis, so the mitotic phase, which is, so this is the growth stages, and then the mitosis phase, which is the division phase. The phases of the cell cycle. So one way to remember them is with the term IPMET. There's actually a C we're going to add to the end, so I sort of joke around that it's IPMET. Interphase, and these are the parts of the cell cycle, not just mitosis. PMAT are mitosis. I is interphase. That's separate from mitosis, but it still is part of the cell cycle. In this one, we're just talking about it. The cell grows. There's two parts to that. They call it gap 1 and gap 2. Growth phases is another way to think of it, growth 1 and growth 2. And then the DNA is copied, and they call that synthesis phase. And so this is the cell looks normal. The DNA is just loose as chromatin in there. It is not wound up into chromosomes. That's why it's not really true to say that the DNA is always in the form of chromosomes, wound up chromosomes. The nucleolus is still there, but not too worried about that. But we will talk about the centrosomes. They're going to play a central part in this. And there's going to be some other parts that you might recognize from when we learned about cells. So here's about interphase. So watch this video. Make sure you take notes just like the other one. Interphase, again, is the life phase. What's happening during interphase? And then we'll move into mitosis. So again, take notes on this.
Because the events that occur in interphase allow mitosis to take place, let us begin our examination of mitosis by taking a closer look at interphase cells. Interphase is defined as the period of a cell's life cycle between one mitosis and the next mitosis, the period of cell growth. As can be seen in this microscopic image of an onion root, some cells are dividing, but the nuclei of most of the cells are round and intact. These are interphase cells. Average human cells, such as these that form connective tissue, spend about 19 hours in interphase and only between 50 to 90 minutes in mitosis and cell division. However, there is considerable variation among different types of cells as to how much of their life cycles are spent in interphase and in mitosis. For example, this human nerve cell rarely reproduces. Instead, it can remain in interphase for up to 60 years. Because nerve cells rarely reproduce, even to replace dead or damaged cells. Nerve damage, such as results from strokes or spinal cord injuries, is usually very serious. On the opposite end of the scale from nerve cells are cancer cells. These purple-stained cancer cells, which stand out clearly against a background of red blood cells, were taken from a leukemia victim. Cancer cells like these do not remain in the interphase state very long. Instead, mitosis and cell division continues at a furious pace. In fact, cancer can be thought of as cells that never rest, that divide over and over again, never remaining very long in interphase. Even though it is often thought of as a resting state between cell divisions, a lot is actually happening inside the nucleus throughout much of this stage of a cell's life cycle. During interphase, deoxyribonucleic acid, or DNA, the enormous molecule that forms the cell's genes and that holds all of the cell's operating instructions, duplicates itself. Deep inside the nucleus of interphase cells, the two DNA strands unwind as new copies of the DNA are created. Scientists refer to this process of DNA duplication as replication, because exact replicas of the DNA are produced. The replication of a cell's DNA makes it possible for two complete sets of biological instructions to be sent into the new daughter cells. During mitosis, DNA is found within chromosomes, but chromosomes don't actually exist during interphase. Instead, DNA and protein form the grainy threads we see here called chromatin. The presence of chromatin is one indication a cell is in interphase, for only with the onset of mitosis does the chromatin mold itself into the distinct shapes of the chromosomes seen here. Scientists still do not understand the exact role that chromosomes play within the cell, but it seems likely that chromosomes are the best way to package DNA, and consequently genes, for shipment into the newly forming cells. The chromosomes seen here, taken from the salivary gland of a fruit fly, provide a convenient material for scientific study because they are 10 times longer and 100 times thicker than normal chromosomes. Working with these cells, scientists have discovered that the dark and light bands we see here are the actual location of genes on the chromosomes. And their studies have also shown that when a particular gene on one of these chromosomes becomes active, the spot on the chromosome where that gene is located takes on a strange, puffed-out appearance. During mitosis, the appearance, movement, separation, and disappearance of chromosomes is of critical importance to the cell reproductive process. In fact, by recognizing what is happening to the chromosomes, we can identify each of the 4 stages of mitosis.
Okay, here's the mitosis. So that takes care of interphase. You're going to want to have an overview stage. You're also going to do a little overview of mitosis. Talk about how it's the reproductive stage or phase of a cell's life. And there's different parts of that. So we got mitosis phases. Interphase is kind of the only separate phase. Some people consider cytokinesis as another phase of mitosis. Some people keep it separate as part of the cell cycle. We'll talk about that one. So prophase is the first phase, and it's going to go into detail on that. But it's one way to remember that is the preparation phase. Then there's metaphase, which is the middle phase. Chromosomes are meeting in the middle, and we'll see what that's like. Then there's the anaphase, which is the apart phase, when the chromatids are pulled apart. The identical copies are separated. And then there's telophase, which is not quite the tears. It's one way to remember it, but it's really not just cytokinesis when the cell splits into 2. This is when it's starting to. It's really the getting back to normal phase, and you'll see why it's called that when we get to that. Okay, so here's prophase. Prophase is when the chromosomes start to form. It's the first time you sort of recognize mitosis is happening, because the chromosomes go from chromatin into these chromatids or chromosomes, those X-shaped chromosomes. So in here, you're going to see a change in the nucleus. Also, these centrioles, centrosomes split apart into daughter centrioles, and they split apart. And here's these spindle fibers. These are actually cytoskeleton. Remember, one of the things we talked about was that both plant and animal cells can have cytoskeleton. It does provide shape, but it does provide movement. Here's the movement here. They're going to work like little puppet masters moving things around. So go ahead and watch the video and then take notes on the video.
The earliest sign that a cell is leaving interphase and is entering the first stage of mitosis, called prophase, is that the chromatin begins to form itself into the definite shapes of separate chromosomes. And at the same time, the nuclear membrane that separates the nucleus from the cytoplasm begins to be absorbed into the cell. As these things happen, the tiny nucleolus found in the nucleus also disappears. Thus, prophase can be defined as the stage of mitosis when the chromosomes first appear, and the nuclear membrane and nucleolus disappear from view. As prophase progresses, the chromosomes become more distinct, and now that the chromosomes can be seen, a rather odd fact becomes apparent: that chromosomes are always found in groups of two, called homologous chromosomes. This means there are two copies of each chromosome present in every cell of the body, with the exception of certain sex cells. In our example, there's one pair of tall chromosomes and one pair of short chromosomes. This state, where double chromosomes are present, is called the diploid condition of a cell. The diploid condition of a cell means that there will be two complete sets of DNA instructions present inside its nucleus, so that if something is wrong with one set of instructions, the other set will still be able to provide the cell with the information it needs to function properly. Under close examination, we find that in early prophase, each individual chromosome, whether tall or short, has a rather thickish appearance. This is because the DNA of each chromosome has replicated itself during interphase. On looking even more closely, we see that each chromosome actually consists of two parts called sister chromatids that contain the duplicated DNA. And at first, the sister chromatids are stuck very close together all along their lengths. It can now be seen that during prophase and part of interphase, there are actually 4 complete sets of DNA instructions present, because the DNA of each homologous pair is doubled at this point. The sister chromatids are held together at a specific region called the centromere. Joined together in this way, the chromosomes begin to arrange themselves so they can separate into the newly forming cells. And as they do so, the sister chromatids become much more obvious. Besides the changes in chromatin and chromosomes, other important events are also occurring inside the cell when prophase begins. In animal cells, a structure called a centriole or centrosome divides into 2 daughter centrioles that migrate to opposite ends of the cell. Between the centrioles, a delicate arrangement of microtubules called the spindle is formed. The microtubules that make up the spindle are called spindle fibers. Spindle fibers are critical to cell reproduction because they help arrange the chromosomes and later in mitosis separate them into 2 equal groups. In normal cells, all the complicated events that define prophase take between 30 and 60 minutes to complete, finally ending when the next stage of mitosis, called metaphase, is reached.
Okay, the next phase of mitosis. So we're talking about the cell cycle, but now we're in the reproductive stage or phase of the cell cycle. Interphase is just the regular life phase. Now we're in mitosis, the reproductive stage. We've already talked about prophase, the preparation phase, as chromosomes form. Also in prophase, the nucleus would start to break apart. In metaphase, by metaphase, the nucleus has dissolved away. So it's no longer in the way. The chromosomes line up in the middle. That's one way to remember metaphase is the middle phase. And you'll notice these X's, they're connected together in the middle. This is an identical copy on one side, identical copy on the other, and they're going to be pulled apart by these spindle fibers, these puppet strings from the centrioles. It's a pretty short phase. Go ahead and watch the video. Make sure to take notes on all videos.
Metaphase, the second stage of mitosis, is defined as the stage of mitosis when all the chromosomes are lined up along the center or equator of the cell. Throughout this short 5 to 10 minute stage, the chromosomes are attached to the spindle fibers, and the centromeres that bind the sister chromatids together split apart. The splitting of the centromeres signals the start of the third stage of mitosis, called anaphase.
All right, the next phase is anaphase. Anaphase is the apart phase, and they really are pulled apart. So they go from being sister chromatids to daughter chromosomes as they're pulled apart. Each of them was an identical copy or a chromosome, but together they've got that. So they're called chromosomes when they're separate, they're called chromosomes when they're together. When they unwind, they should be called chromatin, but they're not going to unwind yet. So we have the sisters pulled apart. It's kind of sad. This is where the sisters, Anna and Anna, are pulled apart to different ends of the cell. So sad. And this is where the DNA really is divided in half. Before this, they had both halves together, both identical copies. This is where the DNA is sort of separated. So we got a full set of copy DNA on one side, full set of DNA on the other side. So watch the video and make sure to take notes.
Anaphase is defined as the stage of mitosis when the sister chromatids separate and move toward opposite poles of the cell. And when this happens, they are no longer called sister chromatids. They are now called daughter chromosomes. This movement happens fairly rapidly, in about 5 minutes, with the result that the spindle fibers disappear from view, and a full diploid set of chromosomes is now found at each end of the cell.
And here's telophase. Telophase is in that page that you saw, that panel where it said they're tearing in two. It's not really. It does start to tear in two. You can see it starts to furrow there. But what's really important, you can't really see it on this, is the nucleus starts to come back and the DNA unwinds. This is not a really good telophase picture. If you were looking for one for your movie, I would choose just a different drawing or draw my own that really shows telophase. It would look like two nucleuses sort of in one cell. These two nucleuses are starting to form, but the cell has a split apart. And the DNA would be back to chromatin or going back to chromatin. So it's going back to that sort of loose DNA that's not tightly wound anymore. The sisters are separated. And one way to remember it is the sisters are far apart and starting their separate lives. They have to call each other by telephone, get telophase. Anyway, go ahead and take notes on the video clip. Pretty fast, quick stage.
Telophase is defined as the stage of mitosis when the new daughter chromosomes change back into the threads of chromatin and new nuclear membranes begin to form. Also during telophase, new nucleoli appear in each newly forming nucleus. As the final stage of mitosis concludes, the cytoplasm divides in half as cell membranes close up around the two new daughter cells. This final process of cell reproduction is called cytokinesis. Cytokinesis is defined simply as the division of the cytoplasm during mitosis. Between 10 and 15 minutes are required to finish both telophase and cytokinesis. When the entire cell reproductive process is completed, the two new cells are returned to the interphase state, and each possesses the identical genes of the parent cell. These two new half-size cells will then go on to grow larger until each achieves the full size of the original parent cell.
And the next part, which some scientists would say this is part of mitosis, but it's not listed as a part of mitosis, but I would be fine if you add it. This is why we got to call it IPMAT-K. The C for cytokinesis is when the cell actually splits into two. So the nucleus is already formed, the DNA is already back into chromatin in here, but now it splits into two. And in an animal cell, it sort of pinches together in the middle, and the cell membranes go back together. But in a plant cell, it has the cell wall. So it actually forms like sort of a little brick layer of this cell wall, and then that divides into two, and the cell membrane would be divided that way. So in plant cells is different. You do need to include that difference in your cell cycle movie. Talk about how cytokinesis is different between plant and animal cells. And that's kind of the end. The next part would be the cell cycle starting over. And we're going back to interphase. These smaller cells now have to grow back up to big cells. And during that time, they'll do whatever cells do, and they'll copy their DNA in interphase. All right, that's the cell cycle. Don't forget to answer these questions. and do your best on them. You'll find the information in this. You can watch the videos again if you need to get more information.